Today I really want to talk about the effect of maternal diet on the epigenome and the implications for human metabolic disease. And I really want to start by just very briefly reviewing the evidence that early life environment is important for future disease risk, then go on and look at the evidence that epigenetic processes are involved, before looking at perhaps ways we can intervene to prevent or reverse these epigenetic changes from occurring. So traditionally then, it's long been accepted that our genes, together with adult lifestyle factors, determine our phenotype and susceptibility to a whole range of chronic diseases, such as heart disease, obesity, diabetes, and cancer. But there is now increasing evidence that early life environment, particularly the prenatal or early postnatal environment, can also affect our susceptibility to these diseases. And the first suggestion that the prenatal environment might be important for future disease risk came from studies from David Barker, who noticed that in the UK, the areas with the highest levels of infant mortality in the 1900s were the same areas with the highest levels of cardiovascular disease 60 to 70 years later on, suggesting that a poor early life environment increased susceptibility to heart disease in later life. David Barker then subsequently went on to show that low birth weight was associated with increased risk of both heart disease and metabolic syndrome in mid to late life, and he saw these associations even within the normal birth weight range. Since then, there have been numerous epidemiological studies which have been carried out throughout the world, and these have basically confirmed the association between low birth weight and heart disease, and also gone on to show that smaller babies have a higher risk of developing hypertension, raised levels of cholesterol, obesity, diabetes. But in all of these studies, low birth weight is thought to be just a very crude indicator of the poor intrauterine environment, which may have been compromised by maternal or environmental factors, including variations in maternal diet. And probably the best example of how maternal diet can affect the health of the child in later life is seen from studies from the Dutch hunger winter, because back in 1944 in the Netherlands there was a period of famine when energy intakes dropped from 1,800 to between 400 and 800 kilocalories per day. And what subsequent studies have shown is that children born to women exposed to famine in early pregnancy have an increased risk of obesity and heart disease, whereas children born to women exposed to famine in late pregnancy have an increased risk of hypertension. So what these studies clearly show is that maternal diet can affect the health of the child in later life, and that the timing of this nutritional constraint is important. So in order to begin to understand how maternal diet can affect the health of the child in later life, a number of animal models have been set up, and these generally involve feeding pregnant rats or mice a low-protein diet during pregnancy or a globally restricted diet. And what you find is the offspring born to such dams develop with features very similar to human metabolic syndrome. So the offspring develop high blood pressure, dyslipidemia, and insulin resistance. So we've become very interested in how this works. So how exactly does alterations in maternal diet affect the development of the fetus, leading to these long-term stable changes in metabolism and physiology? Well, Peter Gluckman and Mark Hansen have suggested that this might be part of a predictive adaptive response. They suggest that during development, an organism can respond to an environmental cue by adjusting its developmental path to produce a phenotype that confers a survival or reproductive advantage. So, for instance, poor maternal nutrition might signal to the fetus that nutrients are scarce. So the fetus will then adapt its metabolism, it will reduce energy demands, increase its propensity to store fat, put less investment in bone or muscle mass, i.e. sort of preparing itself for an uncertain life course. Now, if that organism then finds itself in a postnatal environment where nutrients are indeed scarce, then its metabolism will be matched and that individual will be of low disease risk. On the other hand, if that organism was to find itself now in a postnatal environment where nutrients are actually abundant, its metabolism would be mismatched. It would have this increased capacity to store fat, would be at increased risk of metabolic disease. And this idea of mismatch between the prenatal and postnatal environment has been suggested to account for at least some of the rapid rise we've seen in rates of obesity and diabetes in the developing world when populations move from rural to urban areas or from developing countries to developed ones. But increasingly, there is evidence that there might be a second developmental pathway towards obesity, because increasingly what we're seeing is that overnutrition in early life can also increase the rates of obesity. And certainly now a number of studies have shown that there is a J or U-shaped relationship between birth weight and rates of obesity or diabetes in later life. So babies born at the very highest birth weight are also at increased risk. And again, these studies have been replicated in animals where either high-fat feeding during pregnancy or overfeeding in early life again leads to an increase in obesity. Now, it's unclear how this pathway relates to the mismatch pathway, but what they both suggest is that a single genotype has the ability to form multiple phenotypes, and the exact phenotype derived at depends on the environmental cues that that organism encounters during early life. And there is increasing evidence that the mechanism by which the organism can adapt is through the altered epigenetic regulation of genes. And we've heard that epigenetics is defined as processes that induce stable changes in the expression of genes, with the major epigenetic processes being DNA methylation, histone modification, and microRNAs. So as we've heard, if you have a gene where the CpGs within the promoter are largely methylated, this means that this gene is transcriptionally active. It means that transcription factors can bind and activate that gene. And as we've heard, the way in which transcription factors work is by recruiting histone modifying enzyme complexes to the DNA, because the DNA within our cells is wrapped around these positively charged proteins called histones, which interact with the negatively charged DNA. So what an activating transcription factor does is recruit something called a HAT, which is a histone acetyl transferase, which acetylates the histone tails, neutralizing their positive charge, leading to an opening up of the chromatin. This then allows RNA polymerase to bind and transcription to occur. In contrast, if you have a gene where the CpGs are largely methylated, then this prevents many transcription factors from binding. Instead, you get binding of MECP2, which then recruits a histone deacetylase, which literally removes the acetyl groups from the histones, restoring their positive charge, leading to a closing down of the chromatin. MECP2 can also recruit histone methyltransferases, which then methylate the lysines on the histone tails, preventing their further acetylation, so ensuring that this gene remains closed down. We also know that this methylation is brought about by the DNA methyltransferases, which can be brought to the DNA either with transcription factors or via HDACs or via histone methyltransferases. What we also know is that the methylation status of an individual gene is largely determined during development or early life. So we know that upon fertilization, the methylation marks on the paternal and maternal genomes are largely removed. At this point, we have the differentiation-specific genes held repressed and silenced by the polycomb complex. Then as development proceeds, the polycomb repressive marks are lost, there's an increase in differentiation, an increase in methylation, and effectively we end up with tissue-specific patterns of methylation, and this is effectively responsible for creating and maintaining our different cell types. But once this period of development and differentiation is over, it is thought that these methylation marks are then stable and maintained throughout our life. But what we know is that 1-carbon metabolism, which provides most of the methyl groups for these DNA methylation reactions, is highly dependent upon dietary sources of methyl donors and cofactors. So it's long been suggested that maybe variations in diet during this period when these methylation marks are being set might actually alter the establishment of these marks and might, if you like, reset these marks, and perhaps once reset, these altered marks will then be stably maintained, leading to these long-term changes in metabolism and physiology that we see. So we wanted to test this. So we took pregnant rats, we fed them a control or protein-restricted diet during pregnancy. As soon as the pups were born, they were then given the normal amount of protein. And what you find is the offspring develop with hypertension, dyslipidemia, and insulin resistance. So we wanted to know whether this was due to the altered epigenetic regulation of genes. So we initially looked at the methylation of two genes. These were the glucocorticoid receptor and the peroxisome proliferated activated receptor alpha. And we chose these genes because they are nuclear receptors. They work by activating sets of target genes, and they play key roles in glucose and lipid metabolism, two of the processes which are dysregulated in the protein-restricted offspring. So initially we looked at the methylation across the promoters of these genes in the liver from the control and protein-restricted offspring using a methylation-sensitive PCR assay. And what we found was the level of methylation of both PPAR alpha and GR was reduced in the protein-restricted offspring compared to control. And this reduction in the methylation of PPAR alpha and GR was accompanied by an increase in their mRNA, an increase in expression of their target gene, and an increase in the metabolic processes they control. We've also looked at the methylation of the individual CPGs within the promoters of these genes, and this is looking at PPAR alpha. And what you see again is that the level of methylation, particularly at CPGs 2, 3, 4, and 16, is reduced in the protein-restricted offspring. And in fact, the level of methylation of CPG 3 and 16 predicts 43% and 39% of the variation in PPAR alpha expression in the individual offspring. What we also found is this pattern of methylation in the juvenile offspring was also seen in the adult offspring as well, suggesting that maternal protein restriction can alter the methylation of this gene, and this altered pattern is then maintained throughout the life of the offspring. So how then does this work? How does maternal protein restriction alter the methylation of genes? Well, in order to begin to answer this, we first wanted to address a number of issues. So firstly, we wanted to see, well, how widespread are the changes in gene expression induced by maternal protein restriction, and what types of genes or pathways are affected, and whether the expression changes are persistent, and then also how other nutritional challenges compare. So we took pregnant rats, we fed them a control and protein-restricted diet during pregnancy, and then we looked at gene expression changes in the embryo on embryonic day 8, and then in the liver on embryonic day 14, 18, and postnatal day 84. And what we found was that maternal protein restriction induces the altered expression of a relatively small subset of genes. So in the embryo on embryonic day 8, approximately 3.2% of the genes were changed in the PR embryos compared to the controls. On embryonic day 14, 2.5% of the genes were changed in the PR liver compared to the controls, and in the adult, 1.3% of genes were changed. In terms of the pathways, we found in the protein-restricted embryos compared to controls, there were significant alterations in pathways involved in homeostatic process, development, response to stress, cell adhesion, chromatin modification, and lipid and glucose metabolism. We also saw that very similar pathways were altered in the protein-restricted fetal liver, but interestingly, far fewer pathways were altered in the adult protein-restricted liver compared to controls. And when we compared gene expression patterns in the protein-restricted fetal liver compared to controls, what we found was that only 12% of the genes that were changed in the protein-restricted fetal liver were also changed in the adult PR liver, suggesting that there is a relatively small number of genes whose expression is changed in response to maternal protein restriction, and then whose expression stays persistently changed even in the adult after that maternal nutritional constraint has been removed. And genes involved in this group that are persistently changed are the thyroid receptor, angiotensin receptor, PepC-K, and GR. And in the case of the glucocorticoid receptor, what you find is that you see an increase in GR expression as early as embryonic day 14, which is then maintained right the way through the life of this offspring. But in contrast to genes like GR that are persistently changed, the vast majority of genes that are changed in the fetal liver are only changed in the fetal time point and are not changed in the adult. And genes that are only changed in fetal liver as opposed to adult were a large number of genes interestingly involved in chromatin modification. We also interestingly found a group of genes that were only changed in the adult, and amongst these genes included PPAR alpha. But we were very interested that the fact that maternal protein restriction altered the expression of a large number of genes involved in chromatin modification. So we wanted to see, well, when during development could we detect a change in the methylation of GR? And was this change in methylation then preceded or accompanied or followed by a change in the histones? As understanding when this signal or this change occurred might lead us closer to the signal that was causing this change in methylation. So we've started to look at this using chromatin immunoprecipitation assays. And what we found, if you look in the embryo on embryonic day 8, is that the level of methylated cytosine at the GR promoter is very, very low. You can detect K9 methylation, K27 methylation, which is mediated by the polycomb complex, and you can also detect H3 acetylation, but the levels of these are all equivalent in both the control and protein-restricted groups. If you look in the liver on embryonic day 14, again what you see is the level of methylated cytosine is very low, but what you can detect is a reduction in the level of K9 methylation at the GR promoter in the protein-restricted liver compared to controls. There is also a reduction in K27 methylation at GR in the protein restricted compared to controls, and an increase in histone acetylation in the protein restricted group compared to the controls. And this is consistent with an increase in GR expression, which is what you see at this time point. And interestingly, when we looked on embryonic day 18, what we found was the pattern of K9, K27, and H3 acetylation was very similar to E14. But what we could now detect was levels of methylated cytosine at the GR gene. But what we did see was a decrease in the level of methylated cytosine at the GR promoter in the protein-restricted groups compared to controls. So this suggests that a change in GR methylation occurs around embryonic day 18 and that it is preceded by changes in the histones. So what then about other nutritional challenges? How do they compare? So we compared a protein restriction with a mild global restriction. So this was just a reduction in 30% of overall food intake during pregnancy. And what we found was the pathways changed in the protein-restricted embryos compared to the globally restricted embryos were very, very similar indeed. Although, interestingly, only 11.2% of the genes that were actually changed in the protein-restricted embryos were changed in the globally restricted ones. So despite the fact that there are very similar pathways, it is different genes that make up these pathways that are changed. But what we did see in both the protein-restricted and globally restricted livers on embryonic day 18 was the hypomethylation of GR, suggesting that global restriction, like protein restriction, can alter the epigenetic regulation of genes. So what then about overnutrition? Can that also alter the methylation of genes? Well, Andras Plageman has been looking at this, and he's been using a rat model where he reduces litter size from 12 to 3 on postnatal day 3. And this leads to rapid fat accumulation in the pups and obesity and cardiovascular disturbances in later life. And what he has found is that overnutrition in early life leads to the hypermethylation of POMC. And in particular, what he saw was the hypermethylation of 2 CpGs within the promoter of POMC, and these lie in the binding sites of the transcription factors SP1 and NF-kappa B. And these are absolutely essential for the induction of POMC by leptin and insulin. So in this model, what happens is, despite the fact there are high levels of leptin and insulin, there is no corresponding induction of POMC, suggesting that overnutrition, like undernutrition, can program obesity through the altered epigenetic regulation of genes. So it would seem that there is then growing evidence that changes in early life nutrition can alter the epigenetic regulation of genes involved in controlling metabolism and appetite. So we wondered then whether it was possible to prevent these changes in methylation and metabolism from occurring. So could we intervene? So we took our pregnant rats, we fed them a controlled diet during pregnancy, a protein restricted diet, and then a protein restricted diet supplemented with folic acid. And what we found was that folate supplementation of the protein restricted diet led to an increase in the methylation of both PPAR alpha and GR. It also restored normal levels of PPAR alpha and GR expression and returned levels of beta oxidation and gluconeogenesis to levels seen in controls. And if you look at the methylation of the individual CpGs within the promoter of PPAR alpha, again what you see is that folate supplementation increases the level of methylation to a level seen in controls, although interestingly at a couple of CpGs you actually see hypermethylation. We also compared the gene expression changes in the PR compared to the PRF offspring. And what we found is whereas 1.3% of the genes were changed in the PR liver on postnatal day 84, only 0.7% of the genes were changed in the PRF liver compared to controls, which is consistent with this idea that folate is preventing many of the gene expression changes induced by maternal protein restriction. But obviously there are still a large number of genes that remain changed, including what we saw was a decrease in the expression of the thyroid receptor, which is decreased in the protein-restricted offspring and also in the PRF offspring as well. So if we can prevent then these changes from occurring, can we reverse them? So can we intervene in postnatal life to actually change the phenotype and methylation of genes induced by maternal protein restriction? So we took pregnant rats, we fed them a control protein-restricted diet during pregnancy. Then on postnatal day 28, we fed them a folate-adequate diet or a folate-supplemented diet. 28 days after the end of supplementation, we then looked at the methylation of PPAR alpha. And I should say that we chose to supplement in this juvenile period because we thought that this was a period where there would be rapid growth and differentiation, a period when the epigenome might be more susceptible to environmental change. And what we found was that, as before, maternal protein restriction leads to the hypomethylation of PPAR alpha, but if you supplement with folic acid in this post-weaning period, you get an increase in the methylation of PPAR alpha, and it actually increases above that seen in controls. We interestingly also found that folate supplementation of the control offspring also led to an increase in the methylation of PPAR alpha, increasing it above that seen in controls. And this increase in the methylation of PPAR alpha in the folate supplemented offspring was accompanied by a decrease in expression, a decrease in the level of beta oxidation. And what we saw in both the control and protein restricted offspring supplemented with folic acid was actually the appearance of fatty streaks within the liver and an increase in weight gain. So what this suggests is that firstly it is possible to intervene, that the peripubertal period might be a period of increased sensitivity, but it does also suggest that not all the effects of folate may be beneficial. But we are obviously aware that there has been a great deal of literature on looking at folate supplementation and cancer, and particularly a large number of studies have shown that folate can be protective of a number of different cancers. So we wanted to see whether variations in exposure to folic acid during early life might also alter the epigenetic regulation of genes involved in determining cancer risk. So we've looked at BRCA1, which is the gene which is mutated in inherited forms of breast and ovarian cancer, and it plays a key role in DNA repair in all cell types. And what we found was in the control and protein-restricted offspring, supplemented with folic acid, so these are the filled bars here, you see an increase in BRCA mRNA and protein expression. So folate supplementation increases the expression of this tumor suppressor gene. Now, given that these measurements were being made 28 days after the end of supplementation, well, when levels of folate had returned to control levels, we wondered whether this was due to the altered epigenetic regulation of this gene. So we started to sequence the promoter of BRCA1, and what we found so far is you can detect a decrease in the methylation of 2 CpGs right at the start of, close by to the transcription start site of BRCA1. So this suggests that maybe folate intake during this juvenile period might be very important in determining our risk of cancer. So in summary then, there is growing evidence that alterations in maternal diet can alter the epigenetic regulation of genes involved in controlling metabolism and energy balance. It would appear that these epigenetic periods of susceptibility are certainly both prenatal and postnatal, and we have seen that it may be possible to intervene to either prevent or reverse these altered methylation marks. In terms of folate, we've seen that some of the effects would appear to be beneficial, whereas others are not, and this appears to depend on tissue and the process. And I'd just like to finish by thanking, first of all, Graham Burge, who I have a long-term collaboration with, Emma, who carried out all the PPAR alpha work, Lydia, the BRCA work, and Abby, the array work. Thank you.